So thanks for inviting me to share experience for our program. I'm glad that Jeff did the presentation before about genomic selection and also about the potato breeding program. So that's going to help a lot. Today, the idea is going to be, I'm going to be talking to you a little bit about blueberries to give you context and then the rationale why we do what we do. So I have to teach you a little bit about blueberries before I move actually into the genomic selection part. So first, I just want to acknowledge my team, because I mean, everything that we do is as a team work that we do here, especially with a group that works in the bioinformatics and the genetic quantity and genetic genomics into my program. So today we're going to be talking about southern Highbush blueberry. I mean, that's the model that we're going to be using. And southern Highbush blueberry could not be grown in the south part of the states in the past. And in 1950, my predecessors into this breeding program, they made some crosses of the northern Highbush that we call Vaccinum corimbosum with Vaccinum derowi, a native species of the southeast, to generate what we now they call the southern Highbush. So it's somehow a new varietal type that has low chill requirements and so is able to grow in the south east part of the country, and as well as being able to expand the production of blueberries in tropical and subtropical parts of the globe. And this is probably the only slide that you're going to see about how the plants look like. So I just want to show you how the plants look like. And this is a cultivar that, for example, I released in 2020. It's called Sentinel. And then just to show you the impact of the program can have, in this case, we almost able to yield almost 7,000 pounds per acre. While traditional production in the state produces 2,000 pounds per acre. In the year one of the production. We also do tasting scores. So we test our fruit with large set of tasting panels. And in this case, the tasting score is 27.8. Well, the best scores that we have is around 33 and the worst is around 19. So just to give you context again. The program has been using since the origin, multiple species, and this is to give you an idea, multiple species and multiple techniques to actually make progress into breeding and to selection of different traits. For example, in this case, the use of a wild species Vaccinum elliottii, techniques using chromosome doubling, using coltracine are being used historically in the program. We still use them to make F1 hybrids and F2 back crosses, and then with a couple of crosses, we can recover some of the characteristics that we are looking for. That way, we have been using around 5 or 6 species into the program, into the traditional program, what I call. And in that sense, we keep adding variability and new traits for the consumers. Something that is very interesting that Jeff was just touching, and then I'll be discussing a little bit about that. At the moment that we start working on my predecessor, actually, in 2013, I think he asked me, when I was not leading this program, still he asked me to help him to implement genomic selection into the program. And the first thing that we start discussing is that we needed some pedigree information to find out what are the base information of the program regarding heritabilities and regarding a lot of the inbreeding depression and effective population size, so on and so forth. So this project of reconstructing the pedigree of the program, it took around 4 years since that moment. By the time I was leading this program in 2017, I was able to have a full pedigree. Our pedigree has more than 15,000 lines now, and it goes all the way to the origin of the domestication of the species, which is very useful. Our research program has different objectives, but the ones that we're going to be talking today about is the discover, develop, and apply molecular and genomic tools to optimize genetic gains and accelerate cultivar development. Most of the research part of our program, we do it through students and postdocs as any other public program. But because of this, as a breeding program, and I'm going to give you the experience of a breeder today. I'm not here as a quantity geneticist or genomicist, but as a breeder. We do everything that we need to do for actually to produce better varieties, all the way from field production, genomics, gene editing, and molecular biology. Now, on the other hand, so on one hand, I have the research part of the program, and but on the other hand, I have the breeding program that we manage. And the goal of the breeding program is increase blueberry consumption. And we have a way to do that by developing better cultivars with these different characteristics. Cultivars that are more satisfying for the consumers, but at the same time, they're more profitable for the producers. The program, as I mentioned before, is started in 1950. And before that, we see, we reconstruct also this, this information in one of the publications of one of my students in the past. But we were able to reconstruct all the information that exists for fruit quality, in this case, fruit firmness. And all these are the varieties from the north. And in blue, we have the varieties that have been developed here in the south, or in the University of Florida mainly. And we can see a huge increase in the fruit characteristics. And this is to show you that traditional breeding techniques obviously work. They're just slower, of course. In that sense, the program has released over 100 varieties directly and indirectly. Here I'm showing just a part of them. And then we have a stable pipeline of crosses and selections of different stages, as the same as Jeff was showing before, to make progress and to be able to release 1 to 2 cultivars every year if we need to. With this information, we not only use the cultivars that we release here in Florida or in the Southeast, but also we license our cultivars all over the globe in parts where they can be grown in tropical and subtropical parts of the globe. So once I arrived into this program, the first thing that I wanted to do was training as a quantitative geneticist and breeder. From my background was to estimate the genetic parameters, and usually, you put an experiment in the field, you measure it, and then you try to estimate your remo blabs, traditional remo blabs that we have been using, for many years. I've been training in forestry before working with crops or fruits, and then in forestry, the use of remo blabs, it goes back to 1980s, more or less. So. In that sense, that's the first thing I wanted to do, because I want to understand, about the genetic parameters such as variance, genotype by environment interaction, trait correlations, and so on, genotypic correlations, so on. And with that, we know with blabs, as Jeff taught us before, you can get the predictions and then use this to do breeding value and selection of parents. And whenever you do this enough times, and then you're going in the right direction, eventually you're going to be releasing a cultivar. One of the issues that we have whenever I started in this program is that, and actually whenever we start working with tetraploids or auto tetraploids in the past, in that moment, there was no software to reconstruct the relationship matrices. And that's one of the essential parts that you need actually to calculate remo blabs or to understand your populations. So in that moment, one of the first projects that we got into was to construct a software to construct the relationship matrices. And this was the project of Rodrigo Madeo that unfortunately we lost them as a tetraploid community. Nowadays, the software can construct relationship matrix for any ploidy using pedigree, genomics, or using both, which is called the H matrix. And I think there was a little bit discussion about that. Also, you can construct matrices for additive, for dominance, and also, by use Kronecker products, you can use epistasis as well. There is 13 different methodologies that you can select now in the software to construct your relationship matrix. And this is a package in R that's freely available for everybody to use. After we have that piece, down, was the second issue was, okay, we're working with tetraploids, and Jeff presented before. There is ways to deal with this issue that what dosage parameterization do we need to use in our models? Well, in that time, we're still trying to figure out how to make it happen. So as you see an additive or a dosage model, as I'm showing here, we're asking the question, do we really need to dosage, or we can use other strategies? In that sense, using now having the HGH matrix software that's showing you before, and this analysis within that part, we did it with BGLR, but in reality, we use any software that is available and then fits more needs. For the operational part of the program, we use Ayers Remo, like Jeff was mentioning before. So from that moment, in the past, we start asking different questions that we thought at the same time that probably many of our colleagues working with tetraploid species had the same questions, and that's why we were publishing everything that we were doing. So for example, the effect of the relationship matrix, can we use ratios or continuous genotyping instead of dosage? What happened with non-additive effects, and then ways to optimize genomic selection. In that sense, and also I'm adding the screenshots of the paper so people can go back to them and take a look at them in case they need it. So one of the things that we did to ask the questions of what happened with when you use different relationship matrices is a proof of concept, but at the same time to introduce this idea that you can use marker-based ratio or continuous genotyping, where the ratio of one allele to another, basically the allele frequency of one allele to another. So in that sense, we were able to prove that by using the ratio of the allele frequencies, you can find different similar genetic accuracies or predicting ability than using tetraploid parameterization or diploid parameterizations using molecular markers, and in all cases were better than pedigree data, and obviously much better than not using any pedigree or genomic at all. At the same time, we're trying to optimize the model to find out how many markers do we need, how many probes. We are working with next generation sequencing data or sequence capture. And then some of the questions that we had at that point was basically how many markers we need, how much sequencing depth we need. So fortunately, we had a very large data set of around 16,000 individuals, genotype with over 30,000 markers, and then they were with a high sequencing depth, so we could do all these studies in silico to find out what are the numbers that we need regarding these different questions. We, like Jeff mentioned before as well, you can do genomic selection with a lower marker density that I'm showing here, and I think number of markers we see here, between 2 to 10K is going to work well, but it depends on what are the objectives. If it's purely genomic selection, closer to, between 2 and 5K, you should be fine. But if you want to use other analysis simultaneously with your data, for example, GWAS analysis, maybe you're going to need a little bit more, depending, again, what is going to be the linkage disequilibrium or the effective population size of your populations. With that, then there is different ways that you can use this information. Using information to decrease the cycle of the traditional breeding cycle is one way, but another way is to optimize prediction of parents, as I'm going to show you in a second. The second important thing regarding what we are doing or what we have found with all our research is the use, and I think this is not new again, but this is the information as being coming to us while we do the research. Prior biological information derived from GWAS analysis that we are carrying simultaneously in our program can help with the prediction. In this case, a very simple example, and this is with real data that we have in our program. So we have 2 different populations. Population 1, I believe it was around 900 individuals, and population 2, I think 400 or 500 individuals or something like that. In here, we have different scenarios. So the scenario number 1, and as the different colors, match the plot on the right side. In the scenario number 1 is a cross validation, and then we obtain a prediction ability of 0.49. Scenario number 2, it will be traditional genomic selection without any help of GWAS or other cases. And then we see that when we try to predict from population 1 to population 2, even where in this case, there is some levels of relationship, the accuracy is going to drop, which is expected. You obviously don't expect it to drop below what it will be using traditional phenotypic information or pedigree information. Scenario number 3 is when we use the outcomes of the GWAS analysis, and obviously this is done independently, so which means that the GWAS also is carried out in this population, but then is used in population number 2. So we are able to recover some of this information. And then in scenario number 4, and this for this particular trait, we can show that actually a traditional marker assisted selection can be used. Now, in this case, particular case, the characteristic or the trait that we're talking about is eucalyptol, and this trait is more monogenic or oligogenic, and that's why you see this scenario here. Now, in the paper, we explored many different traits, volatile metabolite traits, and then you can see different patterns depending on the trait that you're working with. Anyhow, the use of prior biological information is always going to help with the prediction. In our program, we do around 100 crosses every year, and as Jeff was mentioning, the optimization of crosses, again, as I mentioned before as well. My background is from forestry, where the population improvement is we paid a lot of attention to that. So given that background, you know, the crossing and the paying attention about the inbreeding and the long term is something that we always do. In that sense, we do around 100 crosses. We use the genotypes from the different stages in the program. So from stage 2, 3, and 4, which I'm going to show you in a second what doesn't mean, but stage 2, basically plants are still in the seminal roots. Stage 3s and 4s are clones, are clonally propagated and put in commercial production sites. And then we use cultivars and wild species. We use all kind of information here. So we have around, in the ground, we have around 2,700 genotypes, apart from the nurseries, you know, and then so that gives us around 4 million possible crosses that we could perform. And then if you think about 100, in groups of 100, you know, you have virtually infinite number of crosses. So as Jeff was mentioned before, and this is an example for breeding programs, so you have the diversity or the relationship among the different a way to calculate the diversity as Jeff was mentioning. This is one of our traits here, translated back from the breeding values to the phenotypic data. And then we have the dotted lines are the average of our population right now. And basically this become an optimization problem. And I'm going to keep saying, as Jeff mentioned, but this is the case. Jeff mentioned about this as well. So in an optimization problem, you always want to be living here in the border, basically. So you want the best of both the scenarios. The higher the trait, but also at the same time, you want to keep higher variability or diversity. In this sense, and because these problems, optimization problems, are very large, you know, you need to try to find methods actually to find this border area where it gets optimized. And then the AlphaMate software, we used that for a couple of years, and it worked very nicely. However, it has a lot of restrictions because I don't think it was made specifically for plants, and then neither for clonally propagated plants like in our case, or perennial plants in our case, like actually we need to. So we have to do a lot of adjustments, play, and then try to trick or fool the software one way or another. At the end of the day, you know, we have to come with a customized optimizer that is the one that we are using nowadays, but it follows the same principles. Somebody was asking about dominance or non-additive effects, you know, and actually I did perform some analysis and work in my PhD back in 2012. I think we published a paper, I believe, in 2014 for diploid species. And then now the question was, and the rationale for this work was, if we do have more allele possibilities in this case, there is a higher chance that you could have more interaction. In that sense, we did test that, and in reality, we find out that when we include dominance effects in the models, you can capture some of that and usually borrows it or takes it from the additive effects, because one of the things that Jeff mentioned earlier, you know, that with additive models, I don't think I'm going to repeat this properly, but the point I want to make is that while additive effects is very important because it's what we use to make the crosses to decide what crosses to make, and that's what is going to give us a prediction of what the performance of the project is going to be. The truth is that if you look into the books and you do the whole derivation of this, there is going to be non-additive effects confounded in the additive models. So while you are able to clean that up, basically, whenever you add the non-additive effects, it doesn't make any difference in the prediction effects. And again, I did the research back in 2012 to demonstrate this. However, again, they might be interesting to add the non-additive effects because you get a cleaner prediction of the additive part, but there is not going to be an increase in the accuracy. And this is for traits that most of the variance is explained for by additive factors, by the way. Now, the largest impact I think that we have found in our program from a point of view of resources, balancing the resources that we have in the program, where do we put them, in reality has been actually in having better genetic information or better genomic information in this sense. So here in this very short paper that I will recommend you to read if you are interested in breeding or being a molecular breeding, we show that using a high quality genome, obviously it helps a lot more to understand the genetic architecture of the traits, but at the same time to reduce the resources that we're putting in genomic, given that we can optimize that as well. Again, just to give you an idea, in the left side you have without the genome before the genome was published. So we're using some draft genomes, and then whenever the genome was published, we were able to sort this out, and also everything changed. Just the number of markers, the number of hits that we are getting. Also, it changed, you know, the genetic architecture of the trait. In some cases, now we are able to see additive dominance and different type of genetic control for the traits. And at the same time, you know, for example, you know, in some cases we have a lot of peaks, you know, we're able to obviously put them where they belong. So again, we also reduce the number of probes because now we can sort this out. Again, we are using next generation sequencing with sequence capture. So we're able to sort out and select where do we want to put the probes. And in that sense, we went from 30,000 original plan that we were originally using to 15,000. And nowadays, actually, we're using 10,000. The use of next generation sequencing or sequence capture in our case, you know, allow us to design whatever we want, whenever we want in reality. In this sense, and in the case of genomic selection, here we can, I'm showing you, you know, that actually in the first case, we're using 30,000 probes, and in the second case, we're using 15,000 optimized probe, and then we are able to achieve the similar predictability with fewer probes, which is going to be translating in fewer resources or on lower cost. In this sense, also, it's important, we are working with the whole community, of course, to try to identify the markers that have been detected by us and also their colleagues to use it as fixed effects, as I was showing you before, that they're going to have impact on the genomic selection. Finally, and this is something that we just published last year, so it's kind of, you know, a summary of everything that we have done and the lessons that we have learned from our program. So again, here we have in light shade the light gray, we have the one population, and in black the second population, and these are the different accuracies that we can obtain across different characteristics in different scenarios. The take-home message here is, in reality, that we know that genomic selection works. I mean, this has been working for different times. You just need to optimize it for your program, is the message. Now, and it works better than using the pedigree information. Yes, even in our case, when we have the whole pedigree history, in most cases, it's going to be better as well. This is our breeding program, and nowadays how we are managing the breeding program. So after we make crosses, we have 20,000 plants in the nurseries, in the first nursery, and from that we reduce to 2,000 plants. And in this case, we use visual selection. Why? Because it's very easy to see, actually, like Jeff was mentioning, if the plant is just producing is one of the selection criteria, and actually the berries are acceptable is the second selection criteria. Well, there is a lot more complex than that, but we get to 2,000 plants, and from that, actually, we clonally propagated, and then we established replicated trials with 200 individuals, 200 clones. And from there, we go to 20 clones that get distributed across the whole state in different commercial farms. And from there, we move to a cultivar. We're using different tools now. The genomic selection basically is happening at this stage here. So we do genotyping of 2,000 plants. Now, probably one question that people ask me all the time is how far or how often do you train the model or retrain the model? In our case, the model is being retrained every year, because as part of being a breeder, we do have to phenotype plants actually to make release of cultivars. And in this sense, so we have a set of individuals that we do phenotype anyway every year, but it's a smaller set. While for the prediction, we can use to improve our prediction every year. Right now in the training population, using the language that Jeff was telling you before, we have around 3,000 or 3,700 individuals. And then we are using that information to run our genomic selection. Very important, genomics by plus pedigree plus the phenotype are going to give you the predictions. In our case, the prediction plus the pedigree we use to generate the cross optimization. The prediction plus the breeder knowledge in reality is what is going to define what the crosses you actually are going to realize out of these optimizations. And then at the same time, what varieties you're going to release. Take home messages, genomic selection works. The challenges, regarding GWAS or genetic correlations, those comes from the crop and the traits, not from genomic selection. Genomic selection is the same as traditional REM of lab that has been used for many years. In our case, we believe that simplicity is the ultimate sophistication. So the use of GBLAB, very simple, translation of what we have been using for many years. The ratio of the alleles is a very simple computation. It doesn't require different, you can use it for any ploidy. Additive models, we're still capturing some of the non-additive effects, and you could clean it with adding the non-additive effects, but we're not trying to predict it in our case. And then low to mid sequencing depths, a difference of what's been that's been told to us in the past that we need to use higher sequencing depth. Crosses optimizations are needed in the long term. And at the, as a breeder in a breeding program, the resource balance is the key. So people, cost, space, and speed. And then Jeff was also trying to exemplify that. How do we put these things together is what it matters. So future, we need to include environmental covariates to improve the predictions, and also phenomics. I think that's where is the key to improve the prediction of our models. And then we're going to keep trying to add biological information into our program as well, into prediction models. Also, we are collaborating with other projects, large projects, to try to improve the genetic resources for the community. And then with that, I will just say, thank you. And I think I just took all my time.